Carbs did not change. Your body did. Not because of anything you chose, not because of discipline or genetics or aging in the way people casually mean. Your body crossed a specific hormonal threshold that quietly rewired how every bite of bread, rice, and potato gets handled. The rules that worked at 45 no longer apply at 62, through no fault of yours and through no fault of the food. But here is what the biology actually shows. The carbohydrates are not the source of the problem. The absence of something that used to manage them is. Women who understand the mechanisms we are walking through today can eat rice at dinner and barely register a spike. Women who do not cut carbs entirely and still watch their blood sugar behave unpredictably, without ever understanding why. Today, we are going through what actually changed in your body after 60, and the exact adjustments that allow bread, rice, and potatoes back on the table without the consequences that made them feel like enemies. Something managed your blood sugar for decades that had nothing to do with diet. When it left, it took the instructions with it. Estrogen was a metabolic regulator throughout your reproductive years, not just a reproductive hormone. One of its quiet functions was keeping your cells sensitive to insulin's signal. Think of insulin as a key and each cell as a lock. When estrogen was circulating, those locks turned easily. Small key, wide door, glucose moved into cells fast, and blood sugar cleared quickly. When estrogen dropped after menopause and stayed down, the locks stiffened. The same key now turns them more slowly, less completely, and glucose that used to clear in 20 minutes lingers for 90. This is not clinical insulin resistance. It is a predictable sensitivity shift that most postmenopausal women were never warned about, because the blood sugar research that established the norms was conducted primarily on men whose hormonal landscape does not make this particular transition. The gap is documented. It explains why a food that was neutral at 42 becomes a problem at 63 without a single other variable changing. And it is only the beginning of the explanation, because there are things you can do, plate by plate, that work around it completely. The sequence you eat your plate in reduces the glucose spike by more than half. Not the portion, the order. Think of your stomach as a gate between a reservoir and a flat field. Open that gate directly to a rush of digestible starch, white bread, rice, potato, and the field floods faster than it can absorb. Glucose enters the blood all at once, demanding a response from locks that are already slow. But fill part of the field first with fiber and protein before the carbohydrates arrive, and the same flood moves slower, spreads thinner, and soaks in without pooling. A 2019 study at Weill Cornell found that eating vegetables and protein before carbohydrates reduced peak blood glucose by as much as 73% compared to eating carbs first. Same food, same total amount, different sequence. The glucose curve did not flatten because less was consumed. It flattened because the rate of entry changed. Fiber physically slows gastric emptying. Protein triggers a hormone called GLP-1 that further decelerates that transit. Together, they reconstruct the buffer estrogen used to maintain automatically, every day, for 30 years, without you thinking about it once. In practice, vegetables and protein land first. Bread comes last. This is actually the architecture behind the plate structure in Strong Past the Drop. I kept running into the same wall with women who understood the mechanism, who knew carbs were not the enemy, and still could not build a week of eating that held blood sugar stable from morning to evening without second-guessing every plate. The protocol maps the exact sequencing, pairing ratios, and meal timing across 8 weeks. Not a food list, a structure. It is on screen if you want it. But there is a glucose clearing mechanism that bypasses insulin entirely, and it has been living in the back of your legs the whole time. The most effective glucose clearing system available to a woman over 60 is not dietary. It lives in the muscle of her legs. Skeletal muscle, particularly in the thighs and calves, contains a glucose transporter called GLUT4. Under normal conditions, GLUT4 waits for an insulin signal before activating, like a loading dock that only opens when authorization comes through. But during and immediately after muscle contraction, GLUT4 moves to the cell surface entirely on its own. No insulin needed. It pulls glucose directly from the bloodstream through a second door that post-menopause never closed and estrogen never managed. Think of blood sugar as water pooling on a floor. Insulin is the mop. Effective, but slow, and slower still when the mop has stiffened. Your leg muscles are the drain. The moment they contract, the pool clears without waiting for the mop at all. A 10-minute walk taken within 30 minutes of eating activates this system reliably, not intensity. Ordinary movement after carbohydrates. And every time those leg muscles are loaded with even modest resistance, the body receives a signal to synthesize more GLUT4 transporters. More transporters means more glucose cleared per stride over time. The walk that managed a moderate spike in week 1 clears a heavier load in week 8. Walking activates the drain. Strength work expands it. The carbohydrate itself, before it even reaches the plate, can also be changed in a way that reduces the glucose demand on the whole system. The same rice cooked tonight and the same rice left in your refrigerator overnight are not the same food. When cooked starch is cooled to refrigerator temperature, the loose, rapidly digestible starch chains begin to reform into a tightly packed crystalline structure called resistant starch. Your digestive enzymes unravel loose starch quickly, like undoing a slack knot. Resistant starch is the tight braid. It breaks down slowly, partially escapes digestion entirely, and feeds gut bacteria instead of entering the bloodstream as glucose. A 2015 study in Advances in Nutrition found that cooling cooked white rice overnight reduced its glycemic response by nearly 50%. Not a different variety, not a smaller serving. The same rice, rearranged at the molecular level by 12 hours of cold. Potatoes undergo the same transformation. Potato salad made from cooled potatoes is a measurably different metabolic event than the same potato eaten hot, even when the carbohydrate content is identical. The cooling step is not aesthetic. It is a structural change in the food itself. And what surrounds that food on the plate changes what the body does with it next. Bread eaten alone and bread eaten alongside fat and protein are not the same metabolic event, even when the slice is identical. Fat slows gastric emptying, the rate at which the stomach releases its contents into the small intestine, where glucose enters the blood. Slower gait, flatter curve. Protein triggers GLP-1 independently, further decelerating that transit. Together, they are the damp cloth wrapped around the carbohydrate. Think of a lit match pressed to dry paper versus that same match held against paper wrapped in a damp cloth. Same fire, slower burn. This is why the same woman can eat bread throughout a full restaurant dinner, olive oil, protein in the main course, fat distributed across the plate, and register almost nothing, then spike on the same bread eaten dry and alone at 7 in the morning. The bread is identical. The context it arrived in is not. Estrogen built that context automatically for decades. Constructing it deliberately, plate by plate, is what the body is asking for now. And not every meal carries the same risk, because the time of day itself shifts the equation. The same carbohydrate produces a harder, longer spike at breakfast than at dinner. The time of day is not the coincidence it looks like. Cortisol peaks in the early morning. This is normal, protective, and unrelated to stress in the common sense. It is the body's wake-up mechanism. It raises blood glucose before the first meal to fuel movement and cognition. But while cortisol is elevated, it actively opposes insulin's ability to clear that glucose into cells. Think of it as a traffic jam at the cellular locks. Insulin is turning the key, but the road into the cell is backed up. The result is that carbohydrates eaten at breakfast produce a higher, longer spike than identical carbohydrates eaten at dinner, in every controlled study that has measured it. This is not an argument against breakfast carbs. It is the reason the morning plate requires the most aggressive sequencing, the heaviest pairing, and the post-meal walk more than any other meal of the day. The morning slot is the least forgiving window. The same 4 adjustments work everywhere. They simply matter most here. Your body after 60 is not a broken version of what it was at 45. It is a body running without a layer of hormonal infrastructure it received automatically for decades. Infrastructure that sequenced glucose, maintained insulin sensitivity, and kept carbohydrates from becoming something worth thinking about. The bread, the rice, the potatoes that sustained human populations for thousands of years. They are still food. They are still fuel for the brain, the legs, the gut microbiome that depends on the resistant starch they carry. They move through a body that now requires 4 adjustments: sequence, pairing, cooling, and movement. Not 4 restrictions, 4 instructions the body is already built to receive. The signal is the same. The infrastructure that received it left with the estrogen. You now understand why the same plate hits differently at 60, and which 4 variables actually govern the outcome. What comes after that is knowing how to execute those adjustments on a real Tuesday morning with a real kitchen and a body still learning its new instructions. That is the gap Strong Past the Drop was built to close. I put it together after too many conversations with women who understood the biology clearly and still could not construct a week of eating that held blood sugar stable, built leg strength, and included the post-meal walk without turning their schedule into a logistics problem. It is 8 weeks. The plate sequencing, the pairing structure, the cooling protocols built into the meal prep, and the walking and leg strength progression designed to expand the GLUT4 drain from week 1 to week 8. It costs something because the structure inside it took real time to develop. The link is on screen.